Okay, so today for the Classics in Immunology Journal Club, we're gonna talk about a real classic from 1960, Peter Nowell, and the title of this paper is Phytohemagglutinin, an initiator of mitosis in cultures of normal human leukocytes, published in Cancer Research in 1960. And this paper is a landmark paper because it started a whole new discipline, which we now know as cellular immunology. And this paper, for the first time, allowed the scientists to take the immune system out of the body and put it into a tissue culture tube or flask or whatever, and look at it under the microscope and fiddle with it and rearrange all kinds of things and so forth. This was the beginning of a whole new era and really set the stage for what I call molecular immunology that started off around 1980. So between 1960 and 1980, this was the paper that allowed everything to progress. Now, to give you some background about Peter, Peter was just a young guy at UPenn, University of Pennsylvania. He was in the pathology department, and he was a cytogeneticist. And his whole thing was that he was trying to look at the chromosomes. Cytogeneticists, they look at the chromosomes under the microscope, and they line them all up, try to see whether or not there's any abnormalities in the chromosomes, particularly in cancer cells. That's what he was all about. He was trying to see whether or not cancer was really caused by abnormalities of the chromosomes and of course the DNA and so forth. At the time in 1960, there weren't very many people that were thinking about along these lines. What happened was that in the process of examining cancer cells under the microscope, he inadvertently included a control group, which were normal human white blood cells. And to his surprise, he found that there were normal human white blood cells that were undergoing proliferation, mitoses. And that was the beginning of the idea in immunology that lymphocytes, one of the white blood cells, were not just sort of boring end stage cells that nobody knew what they did, and they just made up the lymph nodes and the tonsils and circulated it around in the blood and whatever, and nobody really thought. And of course, as we said a couple of weeks ago, Macfarlane Burnett hypothesized that the lymphocytes were really the seat of the immune system, where it was happening. But nobody knew that they could divide. So how can you have an immune response that's based on clonal selection on the one hand and clonal expansion on the other hand if you can't expand? It just didn't make any sense. So this is the paper. And this is where my career began. I didn't get into the business until like 10 years later, but everything I did was really based on this particular paper. In the introduction of this paper, Peter Nowell says that he references another paper that was only in press in 1960. And so I went back and looked at that paper, and that paper basically was a paper that was looking at leukemia cells in culture. And what Peter Nowell was doing in the other paper was in experimental cell research in 1960, was that he was taking blood from leukemic patients and separating out the red blood cells from the white blood cells. And of course, the red blood cells made up like 50% of the total volume of the blood. And so what you would do, what he did is that someone had discovered that there was this extract from plants, and the extract was from the red kidney bean. And that would have, fortuitously, they found that it would clump together red blood cells, agglutinate the red blood cells. And so you take the blood from the patients, throw in a little bit of phytohemagglutinin to agglutinate the hemoglobin containing red blood cells. And as a consequence of that, and then he would let them sit there, and I'll get into the methods, and that would allow him this gravity to separate out the red cells from the plasma, which had all the white blood cells still containing. He was putting these cells, the plasma from leukemic patients, into culture. And as I said, he included a control group from using blood from normal individuals. And what he found was that, and this is where the sort of the serendipity comes in, sort of the word on the street or the gossip is that this was a big mistake on his part. He left some cells in the incubator over a weekend, put the things together on Friday and didn't come back until Monday. And on Monday, he looked down the microscope and he saw, and to his, he knew he was going to find mitotic figures and mitoses in the leukemic cells, but then he found them also in the normal cells. And other people, who I don't know whether this is true or not, this is just gossip. He said that he thought he really had discovered the cause of leukemia at that time, but then he did more experiments and figured out that he hadn't done that. But it was even better than that. I mean, if you can one up that, he found that normal lymphocytes, well, he didn't know what they were. He thought maybe perhaps they were monocytes. He called them monocytes, mononuclear cells, monocytes, and large lymphocytes. If he left them for 3 days, then he could see mitoses, but he didn't see anything on day 1 or day 2. And that was sort of an enigma for his part. And so this paper is about further experiments that he did on the normals, and he didn't talk about the leukemic cells in this paper. And in the materials and methods of this paper, what he would do is he'd take 10 mLs of blood, which is sort of a tube of blood about this big. This would be about 10 mLs. He'd add just 200 microliters of PHA to this blood, and he'd then put it on ice or in the cold room at 4 degrees for just, like 10 minutes. And the red cells would all clump together and agglutinate and fall to the bottom of the tube. And then he could just take a pipette and reach in and take off the plasma. Well, before he did that, he centrifuged the tube at a very low speed for 10 minutes, and that would finish the job of getting the red cells out of the picture. And then he would take that plasma that it now has phytohemagglutinin in it, and he would incubate those plasma in little tissue culture bottles. And tissue culture 199 that we ran into when we were talking about Enders and the tissue culture revolution that happened from his work in 1949. So this is only 10, 11, 12 years later, and nobody really had cultured lymphocytes or white blood cells. If they put the cells into culture, they would sort of, the white blood cells from the total plasma, the granulocytes or the polymorphal nuclear leukocytes, or the microphages, as Metchnikoff called them, would die off within 24, 48 hours, leaving behind mononuclear cells, which were comprised of monocytes on the one hand. About 20% of peripheral blood mononuclear cells are monocytes. And then the other part, about 80%, then are lymphocytes. He would add then penicillin and streptomycin, which was key because you couldn't really culture these things from human beings in the lab very easily without getting them contaminated with bacteria. At the bottom of he put the little flask on its side, and in the bottom of the flask, he would put a microscope slide, and the lymphocytes all the cells would settle down on top of the slide and stick to the slide. Then he could take the slide out periodically. You could set up replica cultures and so forth and take the slide out. And then you could stain the slide, and with the stain that everybody used in hematology and for peripheral blood cells was called Wright-Giemsa, and that was a great stain because it made the white blood cells really stand out from the red blood cells and so forth. And these were the experiments that he did, and this is when he found out that it would take 3 days for the cells to go into mitosis, and it wouldn't happen in 1 day or 2 days. And he found that if he separated the red blood cells, not with phytohemagglutinin in his cultures, but with other kinds of agents that would also agglutinate the red blood cells. One of them was dextran, and another one of the ways to do it, and we did this in the lab when I started out, we defibrinate the blood, and you could put the blood into bottles with glass beads in the bottom, and then you swirl it around, and what would happen is that the blood would start to clot around those glass beads, but it wouldn't form a big clot. The red blood cells and the plasma would stay still fluid phase, and then you could let the blood sit on the lab bench, really, and this is what we always did, and the red blood cells would still come down to the bottom, and you could take the soup off the top. And when he did that with dextran and or defibrillation instead of the phytohemagglutinin, he didn't get any mitoses. So that was key. So that allowed him to then essentially prove that the mitogenic agent in this whole series of experiments was really the phytohemagglutinin. He compared this method with the little bottles and the microscope slide to cultures. He wanted to know whether or not the red blood cells had anything to do with it and the agglutination process and so forth. So there was a way you could culture blood, the whole thing, with in what they called spinner cultures. And you put a magnetic stirrer in the bottom, and you put it onto a stirring plate, and it would keep the blood mixing up so that they wouldn't agglutinate and fall to the bottom, and they would still there. And when he did that, he found that the red blood cells, even when you kept them in suspension, didn't bother the whole thing at all. He still got the mitoses, and so he figured that he could just ignore the red blood cells after that. So the other thing he wondered about was whether or not why did it take 3 days? Why didn't it happen in 1 day, or even less than that? And he wondered whether or not, and he talks about this in the paper, and this is critical for what happened in the next 20 years, essentially. He wondered whether or not there was something in the serum or in the plasma that was necessary for this process to happen, or something that maybe a product that the cells made that the PHA then allowed. From other experiments, they knew that the PHA was binding to the membranes of these cells. And he thought, well, maybe the PHA is opening up pores or something, allowing some sort of a product to get inside and cause the cells to undergo mitosis. And he did experiments where he took after 3 days of culture, when he was seeing the mitoses, he took the medium from that, and he called that conditioned medium. It was conditioned by the growth of the cells, and he put that on new fresh cells, but it didn't make any difference. Still took him 3 days. But now, I mean, after, he basically was wondering whether or not there was such a thing as T cell growth factor before there was such a thing as T cell growth factor, and he was looking for it, but he didn't find it. But I know why he didn't find it, because by 3 days the cells had consumed it all, and we showed that much later, at least a decade or more later than that. 2 decades later. Took us 2 decades to figure this all out. The other thing he did to really pin it onto PHA was that he boiled the PHA before he put it in. That destroyed its activity. And what heat does to proteins is that it makes them all gloppy and gloopy and so forth. So it destroyed any biological activity that depended upon the structure of the protein. So in the discussion of this paper, he makes the point that the PHA effect was a direct action of the PHA onto the cells, onto the white blood cells, and he speculated that it happened because the PHA was binding to the cell surface, and he speculated that it might help something get inside that there wasn't there before that caused this reaction. He then concludes that this whole phenomenon was happening with monocytes or large lymphocytes, and that's what he concludes out of this. They still hadn't come to grips with the fact that lymphocytes could do this sort of thing. So he was hedging at this stage of the game. The final thing is that he continued to be intrigued by this long latent period of 3 days, by the time you added the PHA or set up the cultures until time that something happened. Now, it turns out that in my career, about 10 years later, one of my mentors was a fellow by the name of Ross McIntyre, who was at Dartmouth. And his whole claim to fame was that he was the second guy to show that PHA could cause lymphocytes or white blood cells to proliferate and divide and undergo mitosis. The history of what happened after this particular landmark paper was the very next thing that Peter Nowell did, and he had a paper out then in 1961, was to show that everybody knew at this stage of the game, from the 50s on, that glucocorticoids or adrenal steroids were immunosuppressive, and they would also kill leukemia cells, lymphocytic leukemia cells. Nobody knew why. They just knew that happened. And by this time, by 1960, glucocorticoids were one of the major drugs that were used in chemotherapy of lymphocytic leukemia, and they were very effective immunosuppressants and anti-inflammatory agents. But again, nobody knew why that was. But Peter Nowell published another paper in 1961 showing that what the glucocorticoids were doing, and in any way, was to block this mitosis from happening. And if he waited 24, 48 hours before he added the glucocorticoids, he couldn't block it anymore. So it had to be there in the beginning of the cultures to block this mitotic reaction that was happening 72 hours later. So that was also very key that we'll get into later. The other thing that Peter Nowell did around the same time, this guy was a housefire. He discovered really the first chromosomal abnormality in cancer, which he called, since he was at Penn in Philadelphia, it became known as the Philadelphia chromosome. And that it's a chromosomal abnormality. So this is going back to what he was trying to do in the first place. He wasn't trying to, have a revolution in immunology. He was trying to get into cancer. And the Philadelphia chromosome is due to a translocation of a partial of the arm of one chromosome onto another chromosome, forming a great big chromosome that looks strange under the microscope. It became pathognomonic for chronic myelocytic leukemia. All patients with chronic myelocytic leukemia had this chromosomal abnormality. And later on, much later on, 40 years later on, it was finally figured out what that chromosomal translocation did to cause the chronic myelocytic leukemia. And we'll get into that a little bit later, a lot later. So I went out to dinner with Peter Nowell and I must have been sometime in the 80s. We were at a conference together in a cancer conference together in Houston. We went out to dinner. And so I said to him, I said, well, that must have been great. You're a young guy and you discovered the Philadelphia chromosome, you discovered PHA stimulated lymphocytes and so forth. And he's a very modest guy, very, humble. And he said, yeah, big deal. And then I didn't discover anything else after that. So, whatever, you just should have stopped after 1960. And then it wasn't until we got into the business and after we had gotten into first T-cell growth factor and then interleukin-2 and so forth, one of the very first experiments we did was to throw in glucocorticoids. And lo and behold, because it was easy, it was a simple question to ask, we figured if the growth factor really caused this mitotic thing to happen, then the glucocorticoids were either blocking the production of that factor or they were blocking the activity of that factor. And it was clear cut. They blocked the production of that factor. And then after that, after 1980, and all the rest of the cytokines were being discovered in the 80s and into the 90s, glucocorticoids were basically found to block either the production or the activity of almost all of the cytokines because they block a transcription factor, which is called NF-kappa B, from working. And so in particular, in these times of COVID and so forth, they block the production of the so-called pro-inflammatory cytokines, TNF, tumor necrosis factor, interleukin-1 and interleukin-6. And that's why they are one of the few drugs that have been found that are effective for inhibiting the progression of COVID from sort of asymptomatic into symptomatic and then into the ICU and onto the ventilator, because if they're given early enough, they will block the production of the pro-inflammatory cytokines that cause inflammation in the tiny little air sacs so that the tiny little air sacs fill up with fluid and then you drown. And that's what they gave to our wonderful President Trump, among other things. They gave him some monoclonal antibodies and some putative antiviral drug that doesn't work. So I think the only reason he got better, they really saved this guy, the glucocorticoids. Dexamethasone, it's one of the synthetic glucocorticoids. So I think I better stop there. And seeing you have so many things to look forward to now.
So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter, where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.